Hi everybody. It's taken me a while to get these done because I'm now in a room where I share with 3 other people, but it's pretty awesome. Here's my new boy, Charlie. It's a pretty exciting week because we all got to hold him, and he moved from the incubator out into a regular bed. So he's still going to be in, not sure quite how long, probably at least another week, they say, but depends how he does. And I think when they give them caffeine, they tend to keep them for a certain amount of time to monitor them afterwards. So anyway, he's pretty cute and he's growing fast, and that's all good. Um, I stay in a what they call a parental care room, so I'm here to kind of help out and feed them, and um, they let me stay in one of the beds and give me meals, and so I kind of get a little day trip every day to go and see McCary and Janae. Um, anyway, just thought I'd give you a little update on how things are going, and hope you guys are doing all right. I figured I would do video lessons for this last chapter because um, I already taught you some of the stuff, and if I do the video lessons, then we'll be able to cover the main things for this chapter fairly quickly. So um, we're going to work on chapter 17, and anyway, um, I hope to get your tests marked and back to you by the end of the week so that you'll be able to have them to study with. Good luck, and I miss you. All right, so this is the first lesson for chapter 17, or the first video anyway. I'm going to kind of chunk it up in sections. The first section is kind of explaining DNA structure and DNA replication, things like that. And then we'll get into some gene expression. So chapter 17 is called molecular genetics. So it gets down into the nitty-gritty of the DNA code and how we actually take the DNA code and decode it during transcription and translation. This is just a quote on the bottom that was from your book. I thought it was pretty good quote. It explains how important DNA is. So it says, every aspect of our development, from the first steps of cell division that take place in our mother's wombs to the ability of our muscles to move and voice boxes to talk, are determined by our DNA. So it's important to know a bit about how DNA is expressed. So we've talked quite a bit about chromosomes. You already know that they're long chains of genetic material, so it has the genes in it, found in the nucleus, that when it's condensed, it's visible, and we call it chromatids. Normally, during interphase, it's really thin and thread-like. We call it chromatin at that point, but you can't see it. Just gonna get a little pen so I can mark some stuff. Okay. If we were to look at it really close, so this over here is coiled up. If you were to pull it out, this right here is the DNA, and it's showing over here, okay, the DNA gets wound around little things. So the center is a core of protein, and the DNA is wound around it. Short sections of the DNA is what makes up a gene, so a little segment of it, or section of it. So when we looked at a couple of the drawings last chapter, so maybe from here to here is a certain gene, okay, and each gene codes for a certain trait. That's kind of That's a very basic way of looking at it. Sometimes, like we learned in genetics, there might be a couple of genes that code for certain aspects of a trait, but in general terms, we tend to think of, okay, this gene codes for this trait, or just this gene codes for this trait. Okay, there's approximately 30,000 genes on our 46 chromosomes, so that's a lot of genes. The Human Genome Project was undertaken in order to try and decode all of our genome, and it was successfully completed in 2003. They found that we have about 3 meters of DNA in a given cell. So if we took that and we figured out how many was in our whole body, that would go all the way to the sun and back like 100 times. Pretty cool. Okay, if I can, I will try and find the YouTube videos that I had shown last year. There are certain animations and stuff that are really good, but I did show a little video clip, so if I do, then I'll put the link on the blog and you can watch it from there. And I'll also let Miss Power know so that she can show you the video clip right now about the history. Okay. Basically, you don't have to know a lot about it, but sometimes it makes it a little more interesting. So, DNA was first found by F. Meischer in about 1870, and at that time he called it nuclein. Okay, he saw this jelly-like material and observed it to be nuclein. Watson and Crick are probably the best known for modeling DNA. They were said to have been out at a pub and chatting over some drinks, and they jotted down a bunch of notes on napkins, and they figured out how DNA must have its structure because of all these other studies and other properties that they looked at. So they proposed their model in 1953, but it wasn't confirmed until 1969. And in that model, it said that DNA is a double helix. So helix, if you think of it, is like a slinky. Okay, so if you picture a slinky, this one has two slinkies that run beside each other. So it's a double helix. And if you looked at a ladder, okay, a ladder has two strands with rungs in it. It looks like a twisted ladder. Okay. Each strand is made up of basic units. We've talked about them before, called nucleotides. Okay, so the basic units of nucleic acids are called nucleotides. Each strand, then, of the DNA is a string of nucleotides that are stuck together. Okay, so DNA is one of those macromolecules, or we also call it a polymer. A polymer is a molecule that's made up of a whole bunch of subunits, repeating subunits. Okay, so it's a polymer of nucleotides. A nucleotide in DNA contains a certain sugar called deoxyribose. So that's where the D comes from in DNA. So here's the sugar, deoxyribose. It contains a phosphate group, so here's a phosphate group, and a nitrogenous base. So each nucleotide has these three things. So right beside it is another nucleotide. So here's the sugar, there's a phosphate, and the base. So if you notice, the sugar and the phosphate are exactly the same. What differs between the nucleotides are the bases. So there are 4 different nitrogenous bases found in DNA, and we label them or name the nucleotides by their nitrogenous bases. Okay, so there are 2 purines, meaning they have 2 rings in their structure, like this one up here. Okay, so there's 1 ring, and there's the 2nd ring. Adenine and guanine both have 2 rings. There's 2 that are pyrimidines. You don't have to know which ones are purines and pyrimidines. We're just saying that they have, they're named based on the number of rings in their nitrogenous base. Okay, so when you look at the sequence of DNA, so if you look at this strand over here on the right, you see only 4 letters that make up the sequence. The 4 letters are A, C, G, and T. They're the first letter for each nitrogenous base. So they're named for the nitrogenous base that's found in the nucleotide. When we talk about the overall DNA sequence for a gene, then it's given by the order of the bases A, C, G, and T. And if you have a slightly different version for each gene, like if you have a gene that says, oh, the plant's going to be tall, as opposed to the version of the gene that says the plant's going to be short, then most of the sequence would be the same for the gene, but there'd be slight differences that make it so it has a different height. Okay, this is showing the 4 nucleotides up close. Do you think that I expected to know how to draw these with their full structures? Nope, you're right. Good guess. I expect you to know that they're made up of a phosphate group, a sugar, and a nitrogenous base. Okay. You should know which 4 are the ones that make up DNA. So you'll have to know the letters A, C, G, and T, and you'll have to know or recognize what they stand for. Right. So how are they organized in the DNA molecule? The sides of the double helix, if you look, remember, it's like the sides of the ladder. Okay, so if you look at the sides of the double helix, the things that you find repeating in here are, here's the sugar and the phosphate. Okay, and it keeps going. Sugar, phosphate, sugar, phosphate. So because of that, because it's alternating sugar and phosphate groups, we call it a sugar-phosphate backbone. The steps of the ladder, so what's in here, joining the two, are the nucleotides, or sorry, the nitrogenous bases. It's N-word, silly. So here's a nitrogenous base from this side, P, thymine, and here is the nitrogenous base from this side, adenine. Okay, so they stick out into the middle between the two sugar phosphate backbones, and they're joined by these dotted lines. Okay, so the nitrogenous bases are connected to form the step by hydrogen bonds. So there's a certain number of hydrogen bonds that will join the bases. Okay, they aren't covalently joined, but they're a fairly strong bond in order to hold the molecule together. So you can kind of tell by looking at the drawing which ones, because of the size of the bases, which ones tend to pair up. In other words, which ones are across from each other and fit well together to make the step. A and T, adenine and thymine, are joined by 2 hydrogen bonds. So when you see them, they're like this. Okay, cytosine and guanine are joined by 3 hydrogen bonds. So in order for the DNA molecule to fit properly and be secure and be as stable a molecule as it is, this is the type of pairing that has to happen. And these bases are said to be complementary base pairs. So they fit well together, they hydrogen bond to hold the molecules or the strands of the molecule tightly together. Okay, and again, it's the sequence of A, C, G, T in a given gene that's responsible for whatever that trait is. All right, so the first process we're going to look at is DNA replication. Okay, we've talked about it before, but we didn't really talk about how it happens. We said it has to happen every time the cell divides. So when it's going to undergo mitosis or when it's going to undergo meiosis, DNA replication has to happen, and it's really important that it happens without any errors. So DNA replication is just like it says, a process where one strand of DNA makes a copy of itself. Okay, it happens in the S stage of the cell cycle, so that's during interphase, and it's necessary for cellular reproduction, growth, tissue repair, maintenance. In order to do it, first of all, we have to be able to separate the DNA strands. Okay, so I'm going to show you on the picture to the right. There's a little enzyme here, okay, called helicase. And helicase's job is to kind of break the helix, break the double helix. So it goes through and breaks the hydrogen bonds to separate the two strands of DNA that are making the double helix. So it kind of is breaking the steps, if you think of it that way. So helicase unzips the DNA strand. When it's unzipped, that means that each of the parent strands, so this red one here and this red one here, can then be read by the DNA that is going to make the new strand or the new copy. That enzyme is called DNA polymerase. So if you think about what it's making DNA and it's making a DNA polymer. Okay, it's making a polymer of DNA, so the name of the enzyme is DNA polymerase. Later on, we'll talk about RNA polymerase, and RNA polymerase is the enzyme that's going to make RNA, the polymer of RNA. Okay, so since this is replication, we're making DNA, so it's called DNA polymerase because it's making a DNA polymer. So DNA polymerase reads the old strand and adds free-floating nucleotides in such a way that every A is opposite to a T and every C is opposite to a G, because that's how they like to bond, hydrogen bond. It fuses the free nucleotides together to make the new strand. And it happens until it gets to the other end. Now, it's a little more complex than that because the enzyme only works in one direction, but the way the molecule is built, one strand is in one direction and one is in the other. So you're going to see when you watch the animation a lot more detail. Basically, I would like you to know that helicase is the enzyme that kind of breaks the helix, opens it up. DNA polymerase is going to add the free-floating nucleotides such that A is opposite of T and C is opposite of G. And because of the way it works, it adds them continuously in one direction, but the other side has to be added in little fragments. So the little fragments are called Okazaki fragments, which are then kind of stuck together by DNA ligase. So pretty sure that this link over here will link, if you click on it, it should link to an animation, but if it doesn't, then you can watch the animation. It's linked from the blog. So this one is DNA replication to show you how it happens. And then if you want to pause this and then go watch that, then I'll do a little practice with you. Okay? So if you want to pause it for now, that would be great. Oh, okay. So you should have watched the animation now. This is just a couple of little things about DNA replication. So if you look, the parent strand or the starting strand of DNA is in turquoise. So here's one, and here's the other. Helicase will open them up, and then the DNA polymerase will add the free nucleotides to make new strands, which are in the pinkish red. Okay, so one thing about DNA replication is that half of each newly formed DNA molecule is from the old DNA, and half is from joining the free-floating nucleotides. So since half of it is new and half of it is old, it's said to be semi-conservative. Half is old and half is new. The free-floating nucleotides that are added, they're in constant supply because we break them down from the DNA in the cells of the food that we eat. We know that all cells have DNA in the nucleus in order to control their functions, so this allows them to, anytime we eat plants or animals, which is pretty much all of our diet, then we're taking in DNA, breaking it down. Okay, let's say we had a strand of DNA. I'm just going to do a short one for now. And this was its sequence. So you can write it down with me. Okay, so here is the parent strand. Helicase has opened up and this is the parent strand. I want you to try, see if you can, figure out what the strand would be that's being made. So the DNA polymerase has to add free-floating nucleotides in such a way that they base pair the right way, right? So I want you, I'm going to get you to pause the video for a second. I want you to see if you can figure out what the complementary strand is going to look like. Go. Okay, so I'm going to get, oh, the wrong one. I'm going to get a different color pen. Remember, how do the base pairs work? It's always A makes 2 hydrogen bonds with T, and C makes 3 with G. So everywhere you have a T, the complementary base is going to be an A. Everywhere you have an A, it's going to be a T. Okay, look at you go. G goes with C, C goes with G. G goes with C. A and T, T and A. And so on. Okay, good job. So that is what the complementary strand would look like. And those would be the exact order of the free-floating nucleotides that the DNA polymerase would put. Okay, so again, we're looking at the very basics of this stuff. Just to give you a taste of it for now, you would go into the more nitty-gritty detail of like certain certain sequences that are looked for, certain differences between things like bacteria and our cells, stuff like that. We're doing the basics, okay? So, if you remember back to when we played the game about the cell cycle, it had a proofreading mechanism to make sure that the DNA strand was exactly the way it should be. Okay, so errors in DNA replication, um, our body, our cells, has ways of looking and trying to make sure that there are no errors. Because if there's errors, then it could lead to um, diseases, or it could lead to things that don't work right. Okay, so the errors can occur by substituting the wrong nucleotide, by deleting a step, being missing one altogether, by adding an extra one. Okay, but any of these would lead to mutations. They would be a mutation that can affect the gene or the trait. Some of them would be a silent mutation that don't really affect it at all. Others could be very drastic. So normally we have proofreader enzymes that check the new DNA for mistake. And we have other enzymes that are like biological scissors. We call them restriction enzymes that would cut the mistake out, okay? And then they'd put the right one back in. The correct one would be put in, and it would be glued or stuck or ligated in place by an enzyme called DNA ligase. So I don't necessarily expect you to know much about this, like for the test, but to understand that normally there is a proofreading process where enzymes will come along, one that's going to proofread it, the other that's going to cut it out, and another one that's going to stick the other one in its place. Oh, all right. That's the end of lesson 1, or part 1. You may get to this next part today, but I'm going to stop it there so it's not too big.